Today, let's review Unit 4 of AP Biology on cell communication and cell cycle in just about 20 minutes. Hey guys, this is Mikey from Aevo Prep Academy, and on this channel, we make AP Biology review videos. And this one in particular will review the entirety of Unit 4, which is sort of a shorter unit. In accordance to Campbell Biology, which I like to call the Bible of AP Bio, this unit covers only two chapters, Chapter 11 and Chapter 12. And that's precisely how we'll go about the review today, covering cell communications first, then moving on to the cell cycle. But before we get to all that, if quick summary videos like this one is to your taste, be sure to click that like button, subscribe to our channel, and share it with a friend who needs it more than you do. Okay, so let's begin with Chapter 11. Cell communications is one of the most important sections in AP Biology. I think it has a lot to do with the fact that this is a section that reviews a lot of our understanding of protein structure and function, alongside understanding biological diagrams. So it goes without saying that on the May exam, your knowledge of cell communications, as well as the skills that you learn here, pay massive dividends. So this chapter is largely divided into 5 sections. 1. Introduction to cell communication. 2. Reception. 3. Transduction. 4. Response. And 5. Apoptosis. Yeah, the 5th one is a bit strange to be included here, but it does make sense once you get there. So let's begin with a bit of an introduction first. Whether we're talking about single-celled organisms or multicellular species like ourselves, the ability to communicate is incredibly important. It's pretty obvious why cells would need to communicate and coordinate their efforts in a large organisms like ourselves. But even for single-cell species, it's still important. So let's take a look at Saccharomyces cerevisiae as an example. This is a single-celled yeast species that is commonly used in genetics research alongside fruit flies and zebrafish. And though these are simple yeast cells, they are sexually reproducing with 2 mating types called the alpha type and the A type. Considering how important sexual reproduction is in creating genetic variation in your offspring, alpha cells and A cells need to find each other for, well, you know. But since cells can't literally talk to each other to commence such sexy times like, "How you doing?" they have to rely on chemical signaling instead in order to achieve this feat. So in this diagram, you see that the alpha cells are releasing chemicals called alpha factors, and the A cell, well, it releases A factors. What's fascinating is that the A cells have only the receptors that can bind these alpha factors, while alpha cells only have receptors that can bind A factors. This results in alpha cells and A cells recognizing that each other are nearby, and given that the concentration of where these factors are can be detected by these receptors, cells can orient themselves in the direction toward where there are more factors, eventually finding each other and initiating mating. So with that example out of the way, let's see how this actually works at the mechanical level. Back in the mid-1900s, Earl W. Sutherland experimented on liver cells. He was interested in how liver cells responded to a hormone called epinephrine, specifically in breaking down glycogen to make glucose in these cells. To put this into context, epinephrine is a fight or flight hormone, so breaking glycogen down to put more glucose into your bloodstream, well, is pretty appropriate here. So he discovered that there were 3 major stages to cell communications, at least from the point at which these signaling chemicals were arriving at the cells. So he divided these 3 stages into 1. Reception, 2. Transduction, and 3. Response. And as this image shows, it's almost as if these chemicals are knocking on the door at the plasma membrane, which is then transformed into some biological activity inside the cell through the transduction pathway, which then results in the response. So this is what we're going to do. We'll take each of these stages and dive a bit further in to figure out just what's going on at each of these stages. So let's begin with reception. Reception begins with the arrival of a signal, or sometimes they're called ligands, at the surface of the cell. Now, it is worthwhile noting that these are going to be water-soluble signals that are traveling through the bloodstream or some aqueous environment. Because these signals are water-soluble, though, they can't cross the membranes of the target cells, for all the reasons that we've learned about in Unit 2, by the way. So naturally, the receiving cells have receptors that can bind to these signals to hear that message at the surface of the cells. This phenomenon of a signal binding to a receptor is not all that different from from a substrate binding to the active site of an enzyme. It's the chemical structure and the properties of ligands and receptors, of course, that facilitate their interaction. But here's the most important bit here. If the ligand binds and nothing happens, then no message would be heard inside the cell. I mean, imagine if someone's pressing on your doorbell without texting you first. I know, it's awful. So they're pressing your doorbell, but the chime speaker inside your house is broken or non-responsive. Well, then you wouldn't know that anyone was out there to begin with. So here's what happens. When the ligand binds to that receptor, it causes a change in the shape of the receptor protein on the inside of the cell, which results in some additional chemical interactions to take place. This is what typically results in the triggering of that second stage, which was called the transduction pathway. But before we get too carried away here, let's talk about these receptors a little bit more. In AP Bio, we learned that there are three major categories or types of receptors: G-protein coupled receptors, receptor tyrosine kinases, and ligand-gated ion channels. They're all important, but G-protein coupled receptors take the cake in having appeared on the AP exam the most out of all of the receptors. So let's start there. As the name suggests, G-protein coupled receptors are receptors coupled to a G-protein. So what's a G-protein? Well, let's take a look at this image. Here you see that the receptor is linked to a G protein. So when a ligand binds to this GPCR, the change in its shape results in the removal of GDP that's bound to the G protein. This is replaced by GTP, which is an energetic molecule, and that turns on the G protein, which then migrates along the inner part of the membrane and can now activate downstream target proteins and enzymes that stimulate the initiation of the transduction stage. G protein then returns to its normal position. So it turns out that G-protein coupled receptors are actually very common in biology, with many of them being found in our cell communication pathways, including that liver cell and its epinephrine receptor. The receptor tyrosine kinases work slightly differently. Here we see that there are two monomeric forms of RTKs that, when bound to their ligands, can dimerize or connect together, and that carries out their kinase activity. Well, what's a kinase though? Well, it's just a fancy name to describe a protein that can add phosphate groups to target substrates. So here, with RTKs being activated, they temporarily hold on to these phosphates that they got from ATP and then pass it down to their downstream targets. Well, and that typically activates them. Now lastly, we have ligand-gated ion channels, which, as the name suggests, are channels that can be opened or closed based on whether there is a signaling molecule attached to it or not. But these are mostly seen in our discussion of muscular contractions and other topics that we no longer cover in the AP curriculum, but it's worthwhile knowing what they are. So let's move on to the transduction stage, maybe using that G-protein coupled receptors as having performed that initial reception. Transduction is the second stage of cell communications as outlined by Sutherland, and it involves the passing on of the message from the receptor to whatever target response that the cell is looking to perform. But before we get to that, I think it's helpful to learn about something very important first, and they're called second messengers. Now, second messengers are what directly connects the receptors to the actual transduction phase of the pathway. So typically here, we learn about two major second messengers. The first one is cyclic AMP. As this image shows, it's literally what an adenosine monophosphate that's had its phosphate group cyclically attached to the ribose sugar would look like. You'll almost certainly see cyclic AMP appear on your school test or the May exam, so make sure you note this one down. Now, another second messenger is calcium ions. And while calcium has a significant impact on so many of cells' functions, you're not really responsible for knowing any specific one of them. But you should still be able to infer from passages and diagrams that calciums can act as a second messenger in transduction pathways. So now that we've talked about second messengers, let's just take it from the reception and see how this transduction thing all works out. So first we'll begin with our example of a G-protein coupled receptor. When that ligand binds, the G protein becomes activated with that attachment of GTP. It slides along the cell membrane and activates an enzyme. And this enzyme?
Albert Einstein.
Wait, no, that's a different joke. The enzyme that the G protein activates is adenylyl cyclase, which, as the name suggests, creates cyclic AMP from ATP. So this totally makes sense. We have a receptor that creates these AMP molecules through that G protein and the second messenger, which can now start the transduction process. So let's do just that. In this image, we see that there is this activated relay molecule, and guys, that is cyclic AMP. So you can see that it activates an inactive protein kinase 1 into its active form. But do you remember what kinases were? We said earlier that kinases are enzymes that can add phosphate groups to downstream substrates using ATP. And here we see the beginning of this phosphorylation cascade. So that activated protein kinase 1, phosphorylating and thereby activating protein kinase 2, then kinase 2 doing the same for 3, all the way until you have the activation of the target protein, which would perform some response, is exactly what this transduction is all about. So in essence, the transduction part of the pathway involves a series of kinases activating one another in order to relay that message downwards in the pathway. But wait a minute. At first, this all looks really inefficient. Like, why couldn't cyclic AMP or even just protein kinase 1 activate the target protein at the end of that pathway? Well, the truth is that most diagrams don't really capture this one very important part of transduction pathway, and that is the amplification of the message. Now, what I mean by amplification is like this. See, this protein kinase 1 doesn't just activate one kinase 2, it activates like 10. Then each kinase 2 activates 10 kinase 3, and so on. So instead of thinking about this like a relay race where each runner tags the next one, is that each runner tags 10 runners, and each of those 10 runners then tag 10 more runners each, and then you have this tenfold and in certain cases 100-fold increase in the message each step of the way, leading to a massive number of enzymes becoming simultaneously activated. This is actually better shown in this table. Here we see Sutherland's very own study where one epinephrine molecule binding to its receptor activates the G protein about 100 times, which then results in activations of adenyl cyclase, creating 100 cyclic AMP each time it's been activated. So that's why there are 10,000 of them here. And as you can see, each kinase activates more kinases, and the end result is the production of 100 million glucose molecules from glycogen as a result of a single epinephrine ligand binding to the receptor. Sutherland would go on to win the Nobel Prize for his discovery of cyclic AMP and the general concept of second messengers. So yeah, he's kind of a big deal. So when it comes to response, we don't see a lot of generalizable concepts because responses can vary so much from one ligand to the next. But these responses, as seen in Sutherland's experiment, can be the activation of an enzyme to produce some substrate in the cell. Or it can be the activation of cytoskeletal polymerization for movement, like what we saw with yeast cells, or even activation of genes during a cell's response to its environment, or simply in the process of development. But now that we've sort of talked about each of these three stages, I do want to add two more additional ideas to this whole thing. First, let's talk about our messenger molecules again. We said earlier that we'd be looking at water-soluble messengers, but what about lipid-soluble messengers, like testosterone or estrogen? Well, it turns out that these hormones can easily diffuse through the membrane. So with these messengers going straight into the cell, their receptors actually reside inside of the cytoplasm and are therefore called intracellular or cytoplasmic receptors. But again, once these receptors are activated, they can go on to activate more kinases or other enzymes or even transcription factors to activate gene expression and so on. Now, second is that the transduction pathways can do a bit more than just amplify messages. Because there are multiple steps involved in the transduction pathway, each of those steps can be either enhanced or inhibited to fine-tune the activation of the next kinase. So further, these relay molecules can even branch, leading to multiple responses occurring as a result of a single messenger binding to the receptor. So there is yet another reason that transduction pathways occur through many steps. Now, the last part of chapter 11 deals with apoptosis, which seems a bit off topic, but there is a connection here. So apoptosis, or programmed cell death, is an important part of multicellularity. We know that apoptosis has to occur properly during the development to eliminate cells that don't end up in the mature organism, and we also know that apoptosis can be triggered to prevent cancer cell formation. All of these are really important. Now, scientists have studied apoptosis in many species, but the main model organism that we're looking at today is C. elegans, a very tiny unsegmented worm. In this diagram, we see a normal cell not undergoing apoptosis. It does have a receptor for the death signaling molecule, which we'll see becoming activated in a minute. But inside the cell, here we see that the CD9 protein is active, thereby actively inhibiting CD4. And that's all we see. But when a death signaling molecule arrives at the receptor inhibits CD9, which no longer holds back CD4. So CD4 becomes active, thereby activating CD3, which activates proteases and nucleases, which then ultimately ultimately carries out apoptosis. So there you have it, cell communications. Remember that studying this chapter is not just about understanding the cell communication mechanisms, but also developing critical skills in understanding biological diagrams such as this one and this one, which appear quite frequently on the May exam. Now let's move on to chapter 12 on the cell cycle. This will be definitely a shorter part of the video because most of you, if not all, have already learned about cell division, mainly known as mitosis, at some point in your general biology curriculum. So instead of going into details of mitosis, I will be focusing more on the regulatory aspects of cell cycles. But without further ado, let's get to it. First things first, I should say that this is sort of a turning point in the AP Bio curriculum. Up until this point, everything's been about chemicals or organelles, or just cells doing something at some place and some time. But as we begin our discussion of the cell cycle, we're no longer looking at cells at some static point in time, but rather what happens from one generation to another, meaning that we're really setting up some groundwork for genetics and how those genes manifest real consequences, and of course evolution to come way later. So with that in mind, we can ask ourselves, why do cells divide? Well, for single-celled organisms, cell division is really the act of reproduction. This was sort of the status quo for a long time before multicellularity and sexual reproduction would evolve. But for this course, we typically look at the mechanics and the consequences of cell division in organisms like animals or plants. So we then learn that cells divide in complex species to develop from a zygote to an embryo, and then, of course, to grow once we're born, and even to repair or replace cells that have been damaged. And there's also this specialized cell division called meiosis, which will be on our radar in the next unit. But let's begin by looking at the big picture of the cell cycle first. Here we see that the cell's life cycle starts at G1, proceeds to S, then to G2, followed by the mitotic phase and cytokinesis to start all over again. Now G1, the S, and the G2 are collectively called the interphase. Now G1 stands for gap 1, and it's the phase of the cell where it just grows from having recently divided, sequesters materials, and generally performs its duties. So if this is a liver cell, it might detoxify alcohol, store sugars, and so on. But at some point, typically triggered by size or other signaling systems, the cell would decide to divide. But in order to do so, it has to copy its entire set of DNA. And that happens during the synthesis phase, often known as the S phase. Once DNA has all been copied, we go through several more checks to make sure that we're really ready to divide, be it checking for enough energy and materials to ensuring that this division isn't an error, which all happens in gap 2. Once everything is clear, then off to mitosis the cell goes. Now, of course, the mitotic event involves prophase, prometaphase, now that one might be new, metaphase, anaphase, and telophase, but as mentioned earlier, what happens in each of these phases and the whole DNA stuff, while important, are pretty foundational, so I'll simply link a video in the description below that goes through that stuff in slightly greater detail. Because for AP Bio, it's not so much memorizing the fundamentals that give students trouble, but rather the inferences and application questions layered on top of that basic knowledge that can be challenging. So for the remainder of this video, I want to talk about those topics that delve more into factors that determine the passage through certain phases of the cell cycle and inhibiting mechanisms that may prevent cells from dividing for pretty important reasons. So the cell progresses through its life cycle kind of like how a car moves in drive. Seeing as a lot of you guys are sophomores or juniors who may have just recently begun driving, you know what I'm talking about. When you put your car into gear by selecting drive, the car will naturally start to roll. Now the car can be made to move faster when you press the gas, or it can be slowed down or even stopped when you press the brake pedal. Just like that, we can imagine that cells have a natural cycle that allows it to divide periodically, which can be accelerated or slowed down or even stopped depending on the needs of the organism. So let's talk about that natural cycle timing first. Remember that all of the cell's mechanisms are driven by chemicals, particularly proteins and enzymes activating or inhibiting each other. But it's also worthwhile noting that chemical reactions occur across a certain time frame, with certain reactions taking longer while others take shorter. Why this is so important is that this regularity with which cells divide can be regular due to the predictability of certain chemical reaction rates. So what does this all mean? Well, first, we learned that there is a very specific molecule inside our cells called cyclin. But as we see in this diagram, if we were to start tracking what's happening from G1, cyclin is largely degraded. But just as mentioned earlier, there is a specific amount of time it takes for chemical reactions to occur. And in this scenario, we see that cyclin can reconstitute itself, and that's a chemical reaction that we're talking about, which would occur across a certain period of time. This is significant because as cyclin becomes more and more concentrated inside the cell, it begins to activate a target protein called the cyclin-dependent kinase, which, as the name suggests, depends on that complete cyclin to become activated. But it's also a kinase, which, as we talked about in the previous chapter, has the capacity to attach phosphate groups to target proteins like enzymes. The attachment of those phosphates, well, that potentially activates those target enzymes, and this combination of cyclin and cyclin-dependent kinases, which are called maturation promotion factors, can trigger a bunch of events that eventually lead through the passage of these phases, which in this case is the mitotic phase. Now, cyclin is then broken down and the whole thing starts all over again. So it's worthwhile mentioning that the example that we've just demonstrated is one of several cyclin-driven pathways that can take cells from G1 to S to G2, and so on. But given that cyclin has some regularity with which it takes cells through these several stages, this sets up the drive mode in a car where the car just moves at a constant rate. Now, let's talk about how we can either accelerate or inhibit cells' passage through the cycle. Now, these mechanisms that speed up or inhibit cell cycles are typically thought of as being external signals. This, of course, would be in contrast to cyclins, which were internal to the cell. So let's take a look at something that can speed up cell division. Here in this diagram, we see something called platelet-derived epidermal growth factor, which is essentially a protein that can diffuse through our bodies and stimulate cell division. This is essential for repair, like if you've been injured. PDGF would help accelerate blood vessel formation and cell division. With a culture that's been treated with PDGF, you can see cell division, but without it, you don't really. So while this image doesn't show you what's actually going on inside of the cell, you can sort of infer from your knowledge from chapter 11 that these cell divisions are being stimulated because the signal transduction pathway that are turned on when these PDGF molecules land at the surface of the target cells. But what's more important for AP Bio are factors that can actually inhibit cell division. And I'll tell you the reason for this right up front. When a cell that is not supposed to divide divides, that's what we call cancer. And while there are so many possible things that could have gone wrong to produce a cancerous cell, one of the major features of any cancers is uninhibited cell division. So let's take a look at these factors that are supposed to inhibit cell division. The first is called anchorage dependence. This simply means that unless cells are anchored in a place where they're part of a tissue, they won't divide. This is sensible because you would not want some random cell floating around in your vessels to start dividing and cause a blockage. The second, and one that links a bit better to the course, is called density-dependent inhibition. Here we see that cells will not divide if they recognize that there are cells around them in higher density, like in an organ. For example, your kidneys don't continue growing bigger and bigger throughout your life, and density dependence has a lot to do with that. But the reason that this factor is important for the course is that we learn how membrane-bound proteins on adjacent cells can interact, thereby sending signals to the interior of each other to inhibit that cell division. So diagrams like this comes out quite often to link chapter 11 and chapter 12, so it's definitely worthwhile looking into this a little bit more. Alright, so that about sums up the major points of Unit 4 covering cell communication and cell division. Hopefully you have a better understanding of what's going on, and if not, you can either comment below or post on Reddit at r/APBio, where I sometimes hang out. And you can of course watch those videos in the description below. As always, I'd really appreciate it if you guys can like this video and subscribe to our channel, as it helps us tremendously with the algorithm and helps us reach more students like you around the globe. This has been Mikey with Aevo Prep Academy. We'll see you in the next video.